Hello everyone, I'm Eber. Today we will be examining to see if 9 is really the optimal pH for the enzyme trypsin. So you might ask yourself, what is an enzyme? An enzyme is a catalyst that lowers the activation energy needed for reactions to occur. It does so by forming the bonds of substrate molecules and creating microenvironments that promote the reactions. Most enzymes are globular proteins, and you begin the catalyzation of reactions when an enzyme binds to a specific substrate molecule at an active site. This binding creates conformational change in the substrate molecule that in turn lowers activation energy. This presence of enzymes is so critical to human life because without them, the chemical reactions in our body would not proceed in a fast enough time to meet the needs of human life. This is largely due to the high activation energy chemical reactions in our body require. For this experiment, I hypothesized that as the pH deviates from the optimal pH at 9, we will see a decrease of enzyme activity during the breakdown of azocasein. Studies have shown that the optimal pH for the enzyme trypsin is around 8, but prior research I have done shows its highest activity at pH 9. For the second round of enzyme labs, I really wanted to dive into this and make it a more valid point. I also know that if enzymes aren't at its optimal pH, it can affect its conformation, which then affects its binding to substrates. This ultimately led me to my question of what would happen to the trypsin activity if the environment deviated from pH 9. The purpose of my experimental design was to test buffers at different pH levels ranging from 7 to 12 and to examine how enzyme activity changes as it deviates from pH 9. I first started off by gathering 5 1.5 milliliter clean centrifuge tubes. I then filled the 5 centrifuge tubes with 450 microliters of 0.15 molar sodium chloride 20 tris ECO to achieve a buffer at pH 7. I then added 500 microliters of azocasein and 50 microliters of the enzyme trypsin into each of the 5 centrifuge tubes. I then incubated the 5 tubes for 10 minutes at 50 degrees Celsius. After incubation, I inserted 500 microliters of 10% TCA into each of the tubes and mixed the solution. I then centrifuged the tubes at 14,000 grams for 2 minutes. I carefully transferred the liquid portion, otherwise known as supernatant, of the centrifuge tubes into 5 different cuvettes. Once the cuvettes were prepared, I cleared the spectrophotometer using deionized water and set it at 440 nanometers. I then inserted the cuvettes into the spectrophotometer individually. I recorded the absorbance for each of the cuvettes at 440 nanometers. I then repeated steps 1 through 11 for, for each of the buffers at pH 8, 9, 10, 11, and 12. The concentration was kept the same at 450 microliters of buffer and the same for 500 microliters of azocasein and 50 microliters of the enzyme trypsin. It is important to have a control variable without the enzyme trypsin. Trypsin is an enzyme that breaks down the dietary protein. In the case of this lab, the dietary protein will be azocasein. There are cases in which a protein will begin to break down without the assistance of an enzyme. It is critical to have a sample without trypsin because it shows the scientist if azocasein undergoes a breakdown without the presence of trypsin. It will allow the data to be more accurate because it will inform the scientist if the enzyme is the only factor in the breakdown of azocasein or not. The control samples were created and went through the same process as stated in the procedure in the last slide. The only difference is that the 50 microliters of trypsin is replaced with 50 microliters of deionized water for all the same. So what we have here are the averages of our control samples. As we can remember, none of these samples contain any azocasein. The azocasein in all these samples is replaced with 50 microliters of deionized water. But as we can see for pH 7, 8, 9, 10, 11, and 12, it undergoes some type of breakdown, which indicates that azocasein does undergo natural breakdown without the use of an enzyme. So in the next slide, you will see how we use this data to get the true values of the breakdown of azocasein. So you might ask, what am I going to do with the control samples? Well, first you would take an original trypsin containing sample and subtract it against the average of the control sample at that certain pH. And when you do this, it will give you the true value for your sample. And I can do this for any of the samples I use in this experiment to get the true value for the trypsin containing sample. So what we have here are the results for our trypsin containing samples. As we can see, this graph represents a relationship between product produced and pH 7, 8, 9, 10, 11, and 12. We can see that as the pH increases to 9, there is an increase in product produced, which indicates an increase in enzyme activity. But after pH 9, we can see that there's a drop off in product produced, which indicates a decrease in enzyme activity. If we look closely, we can see that none of these standard deviations overlap, which indicates that all these data sets are statistically different. So in other words, what that means is that pH 9 is indeed the optimal pH for trypsin because it is a pH that produces more product, which indicates that trypsin is the most active at pH 9. Therefore, my hypothesis is proven because pH 9 is optimal pH for the enzyme trypsin. So from the data on the last slide, I concluded that none of the data was significantly similar because none of the standard deviations overlapped. But I felt that relying on the human eye is not as accurate. So I decided to conduct a t-test in which it analyzes two sets of data and analyzes to see if the data is significantly different or significantly similar. And I know that if the p-value is less than 0.05, then the alternate hypothesis is accepted. And what that means is that the hypothesis stated in the first column will be rejected and the opposite will be accepted. So in this case, I analyzed pH 9 and pH 7, and that was significantly different. I did the same for pH 9 and pH 8, pH 9 and pH 10, pH 9 and pH 11, pH 9 and pH 12, and all those data sets were significantly different. So what this indicates is that pH 9 is the pH with the highest enzyme activity, and that no other data sets has data similar to this. So this indeed means that in the last slide, pH 9 is the pH and is the optimal pH for trypsin. I would say that my experiment was a valid method to test my hypothesis. It provided me with the necessary data to prove at what pH the optimal pH was present at. In this experiment, my hypothesis was proven because it proved that the optimal pH was located at pH 9, as stated in my hypothesis. The visual representation presented on slide 8 shows that the pH at 9 was the highest with enzyme activity. I went a step further and conducted a t-test which proved that all my data was analytically significantly different. This further proved that in this experiment, that pH at 9 is the optimal pH for trypsin. It is very important to have a pH that is optimal for all the enzymes in your body. For example, amylase is an enzyme in the mouth that breaks down starch. It is ineffective at low pHs and it is denatured at high pHs. This is why it's important for its environments to be the optimal pH. If I were to change something for the next experiment, I would test more specific pH values. Now that I know that the optimal pH value is at 9, I could use this and look at what decimal value of 9 holds the most enzyme activity. This would allow my data to show a more precise pH with the highest possible rate of enzyme activity. The pH in this experiment is too broad, and having a more specific number can yield more accurate data. An error that could arise in my data was when centrifuging my samples. In this lab, I only centrifuged the samples one time regardless of what the solution looked like. This might have not been enough to completely separate the supernatant and the pellet. This could have led to inaccurate data when my samples were read by the spectrophotometer. I could eliminate this error by recentrifuging samples that do not look completely separated. Before we conclude, I want to thank Dr. Margolis for his advice during this experiment. I want to thank James Stegman for his PowerPoint statistics that helped with the t-test. And I want to thank the faculty at the Department of Biology at Towson University for providing the laboratory manual. Thank you for coming to my presentation.